Dear students, today we will be discussing about cloning and recombinant DNA technology. And this course or this lecture actually entails the basics for this pharmaceutical biotechnology, because all these biopharmaceuticals or the pharmaceutical or recombinant products are actually produced due to these technologies, which is actually this technology which is called as recombinant DNA technology, or the cloning efforts which we could do. Uh, until unless we are not expert in this cloning and recombinant DNA technology, we can't produce a recombinant protein or a biopharmaceutical. So to be well versed, we have to be very knowledgeable about this all things, cloning and the recombinant DNA technology. Let's move further. The overall objective of this lecture is to understand what's recombinant DNA technology is and what are the different related terms uh, uh, to cloning and recombinant DNA technology, as well as to realize the importance of recombinant DNA technology and to learn how different elements of an expression vector works together to express a certain recombinant protein. So starting with, if we go quickly through this, so you all know, have seen this famous insulin product, which is actually Humulin R. It's a regular Humulin. How this is produced, let's go through it. That we have a expression vector, which is a plasmid basically, and it contains a EcoR restriction site here. What we do is that we restrict this plasmid with this EcoR1 enzyme, and on the other hand, we have a foreign DNA or the DNA of our own interest or the gene of interest, which we need to be, in which we are interested to replicate as well as express that foreign gene. This could be a growth hormone, this could be insulin, as in this case. And what we see is that in this foreign DNA, we have restriction enzymes or palindromic sequence for this EcoR1 site here as well as here. What we do is we restrict this foreign DNA with the EcoR1 along with the plasmid. We also restrict this plasmid with EcoR1 here. And what it produces is that it would produce sticky ends both in the plasmid as well as here in the EcoR1. We will go through the sticky ends, that how these restriction enzymes work in the future next slides. But to be very precise here, that these foreign particles would integrate into this or would be ligated into this plasmid, and we would have a recombinant DNA plasmid here. And this DNA plasmid would be delivered to the expression system. This expression system is actually the bacteria in which we want to express this plasmid, and this plasmid would be transferred back into the bacteria, and then those bacteria would be cultured to express this protein, and later on we will isolate this protein from the bacteria and the media, purify those proteins and make a formulation and pack it into a vial, which is normally used by the humans and named as Humulin R. If we go through the steps, there are actually 5-6 steps which are involved in the production of this Humulin. First is we have to generate our foreign DNA, that how the gene of interest, how we isolate this gene of interest. So basically what we normally do is we actually do a PCR to amplify this gene of interest from the DNA of our interest. Normally what happens is that whenever we should know it, that the gene of interest should be only the coding portions. We all know it that in eukaryotic system, we have exon and introns in the gene. So normally how we amplify this gene of interest is, what we do is we design primers against the gene which has at the ends, we have those restriction enzyme sequences through which we want to cut that the product to be ligated back into the plasmid. What we normally do is say, for example, insulin is produced in the pancreas. So what we do, we total extract the mRNA from the pancreas, we convert it into the cDNA and use that cDNA to amplify the insulin gene from that cDNA. We all know it that cDNA or that mRNA would be having the complete sequence of the gene of insulin without the introns. That would be only composed, that mature mRNA would be composed of only the exonic portions. And when we amplify that with the primers having the restriction enzyme sequences at the 5' end of those primers, we would amplify whole of that gene in the first step. That is actually called as the isolation of the gene of interest. On the second hand, we would integrate it into the expression vector, which is actually the vector which would be restricted with the same enzyme which is present at the ends of that PCR product which we have amplified, or the foreign gene. We would integrate it into the plasmid vector, and then after we would insert back, or we will do a transformation of the bacteria with the foreign plasmid which we have, or the recombinant plasmid which we have made. And after that, we will grow that cells, grows of bacteria which are actually involved, which we need to be replicated. And after application or culturing, we would be in a position that we will culture that, then isolate the protein and purify the protein, and then the last step is that we formulate a product. We formulate a product to be delivered to the human consumption. So this is how we actually use these things, recombinant DNA technology, to produce it's the same procedure. We will move quickly from this. Now comes the point how we isolate the gene of interest. The gene of interest is a small segment in the large DNA molecule, and we actually use, as I told earlier, that we normally use the PCR to amplify those segments, and those PCR primers which are used by us actually contains the enzyme restriction enzyme sites at the end of those 5' ends of those products. And we have restriction enzymes which cut the double-stranded helix at specific sites, and these specific sites are actually called as palindromic sequences, and every restriction enzyme has a different palindromic sequence. Let's go into the details what are palindromic sequence and how they work on. So these are few examples. There are different types of palindromic sequences. One which could produce sticky ends, and the one which could produce the blunt ends. Basically, restriction enzymes could be divided into two types: which produce blunt ends, and the other which produce the sticky ends. So normally, if we give an example of HEPA1 or SMA1, they actually cuts the DNA from the center and they produce a blunt ends. While if we take examples of EcoR1, Hindi3, PST1, they actually produce 5' or 3' overhang or sticky ends. If you see these sequences, it's GTTAC, and on the inverse, it's on the reverse side, it's again GTTAC. So such a sequence is actually called a palindromic sequence, which is similar to each other when we read it from 5' to 3' on both the ends. If you see similarly, if you see Hindi3, it's AA GCTT, it's AA GCTT, and we could read, if you read from the 5' end, it would be similar sequence, and such a sequence is called a palindromic sequence. Let's move further. So basically, enzymes with global cuts give sticky ends. If there are certain enzymes, as I told earlier, we divided into two types: the ones which produce sticky ends, one which don't produce sticky ends, other they produce a blunt end. So here is an example of Hindi3, which actually produce a 5' overhang. And if you see, it's 5' and there is a 5 overhang, and this is 3' and again this is a 5 overhang, while KPN1 leaves a 3' overhang that it produces a long extension at the 3' end. Similarly, here is the again this produce a 3' overhang. If we move further, so there are some enzymes like HEPA1 is SMA1 which actually produce a blunt end, and that it cuts from the center, like here it cuts from the center, and these are difficult to ligate again, but ligation is possible, but to a limited end. Moving further, yeah, this is too interesting to understand. This is to make you understand how restriction enzymes products are like restricted. Here they have shown an example that we have a fragment of 4 kb DNA, and if we cut initially it with the Hindi 3, so 4 kb at this point, it would, this point is almost 2.8 kb from this 0, so it would produce two fragments from 0 to 2.8 and 2.8 to 4. So basically it would produce two bands from 0 to this size and this, and when we run it on the gel we will be having two bands, one is 2.8 kb and other is 1.2 kb, and if you sum both these two it would be 4 kb. Similarly, if we cut this 4 kb fragment with BAMH1 and we consider it that BAMH1, there are two restriction sites for BAMH1 in this 4 kb fragments at 1.8 kb and at 3.1 kb. So we would be having three fragments of the DNA if cut it at two restriction points. One is from this point to this point, other is from this point to this point, and the third is from this point to this point. And when we run it on the gel, then we would be having a 1.8 kb fragment and 1.3 kb fragment from 1.8 to 3.1 and the 0.9 kb from this. So we would be having three bands if BAMH1 cuts this 4 kb fragment at 2 points. If we do a double digestion, both with Hindi 3 and BAMH1, then certainly the restriction pattern would change. And we would be having a restriction pattern like this. If you see, we would be having a 1.8 kb fragment from here. We would be having a 1 kb fragment from this to this. We would be having a 0.9 kb fragment from this to this, and 0.3 kb fragment from this to this. So there would be 4 fragments if we restrict this 4 kb segment with two enzymes, that is Hindi 3 and BAMH1. So this is how the restriction enzymes would restrict, keeping in view the palindromic sequence available in the DNA which is being restricted. Now coming to the DNA ligases. So how DNA ligase work? DNA ligation is actually the act of joining together DNA strands with covalent bonds with the aim of making new viable DNA or plasmids. So basically we have a T4 DNA ligase which has the unique ability to join sticky and blunt ended fragments. So if you see here, these are complementary bases, and if they fit it here, so DNA ligase would ligate this last base here to help the ligation and complete the double stranded structure. While in terms of blunt ends, this also works. Sticky ends that are complementary can be ligated together, even they are produced by different enzymes. Means that if the sticky, we are using two different restriction enzymes, but the overhang which are produced either 5' or 3', they have complementary bases. Certainly they would ligate together, but the palindromic sequence would be disturbed, and in future there would be no restriction site available at this point. While if there is no complementary between the sticky ends after restriction enzyme treatment, then there would be no ligation taking place between the two complementary strands. So we have to be very sure that we are using appropriate restriction enzymes. Just here, you take an example that BAMH1 and BGL2 produce such sticky ends that could have the complementary with each other, complementarity with each other, and they could ligate together, but there would be no palindromic sequence available for future, and BAMH1 or BGL1 BGL2 can't restrict this restriction site in future. Just have a look here that there is GATC, and again there is a GATC with the restriction, but the last base here is A, here is G. So they could have the complementarity. They have the complementarity and they could ligate, but when this ligation takes place in future, none of these enzymes could restrict this in future. So this restriction site would be lost for the future use. Now, DNA ligase can also join blunt ends. Certainly we know it. And for this reason, DNA fragments with blunt ends generated by different enzymes can be ligated together, and but with a lower efficiency, and usually cannot be recutted by either original restriction site even, restriction enzyme even. So what happens is, say for example, here there are two enzymes, SMA1 and RA1, which actually creates blunt ends, and triple C, the triple G, triple A, triple T. These are the palindromic sequence which are specific for these two restriction enzymes. And we have four possibilities once we join them, that we could have the original sequences of these enzymes joined together, but certainly these would not be recombinants. Or we could have recombinants in shape of triple C, triple C, triple T, or triple A, triple G. And once these are obtained, ligation of reconstituted SMA1 and RA1 mixed products would end to that we would not be able to recut these ligated products, which is these two, in future with the SMA1 and RA1. Moving further, that what are plasmids? You all know it, that plasmids are actually extra chromosomal DNA this finally muted. I can't find anyone who is. Kindly mute this. I can't find anyone who's okay. We were discussing about plasmids. So you all know it, that plasmids are actually extra chromosomal DNA, and previously you are knowing it that plasmids are actually, they transfer the antibiotic resistance between the bacteria, and F plasmids were known to be for this. And if we see example, say here, this is the F positive cell, and through sex pili actually it's transferred its plasmid, replicated plasmid, to the other cell, and in this way both become F positive cells, and you have a resistance capacity in both the cells. So now moving further, that what's the structure or genetic structure of the plasmid? So we have two types of plasmids. One are called the normal replicate plasmids, and other are called the actually the expression plasmids or expression vectors. So we have normal vectors and we have expression vectors. Expression vectors are such vectors in which you can express your transgene or the gene of interest. So if we go through the structure of this expression vector, which is actually PUC18 vector, so we have a amplicin resistance gene. It's a selection marker, and this actually helps while selecting the plasmids which carry the transgene. And we have an ORI, which is actually origin of replication. We have a lag promoter, and we have a lag Z complex which is carry a multiple cloning site. This multiple cloning site is actually also called as polylinker and contain sequences for multiple restriction site. So basically, this amplicin resistance is actually a selectable marker when expressed on a plasmid, which allow host cells to survive in ampicillin medium. So, going to a little detail of these all elements which are present within the expression vector. So one is the multiple cloning site, and a short segment of DNA which contains many up to 20 restriction sites for several restriction enzymes is called a polylinker or a multiple cloning site, and we can use such a restriction enzyme which is not present in all of the rest of the DNA of the transgene as well as the plasmid DNA, so that our DNA would not plasmid DNA don't get restricted. Rather, we have a specific restriction enzymes in this multiple cloning site. Moving to the other part is the promoter. So whenever you are expressing a transgene, it depends what type of transgene you are expressing. Either you are expressing a prokaryotic transgene, or you are expressing uh, a eukaryotic transgene or a prokaryotic transgene. So whenever you are using this, you have to be very sure that you have to, if you are expressing a eukaryotic transgene, you have to be very vigilant that you are using a eukaryotic promoter. Unless you don't have a eukaryotic promoter, that eukaryotic transgene would not express. So normally eukaryotic promoters are different from the prokaryotic promoters, and eukaryotic promoters we have from the initiation. This is the initiation mRNA start site, and below that -10 bases के ऊपर we have a Tata box and a cat box, and these are actually the points which are recognized by the transcription machinery, and these transcription machinery later on recognizes these promoter respective elements and start the transcription. Just have a look here that these are the enhancer sequences. This is the promoter sequence and this is the transcription site. And what happens is the RNA polymerase attaches here, and along with the different mediators and activator proteins which are attached to the different enhancers and the data and the other cat boxes which we discussed earlier, would recognize this transcription initiation site and would start transcription from here. So promoter is actually the one which starts the transcription, with help in recognition and starting of the transcription. Now the other point is the most critical is the lac operon. What's lac operon? From the name you could understand that lac means lactose and operon is the functional structure of that DNA segment. Which means that which is involved in the metabolism of lactose. So this lac operon is actually a segment of DNA that includes promoter, an operator, and 3 structural genes which code for lactose metabolizing enzymes. We all know that our bacteria actually takes energy from the glucose, but in absence of glucose, it takes the lactose from the outside of the bacteria, which is actually extracellularly available lactose, and bring it inside, bring that lactose inside into the cell, and then metabolize that lactose into glucose to be used as energy source. So if we go through the structure of this lac operon, it has a regulatory promoter here. Then there is a regulatory gene which is actually an portion for the repressor protein, then there is a promoter for structural gene, then is a operator sequence, then we start have a structural genes. There are three structural genes, that is LAC-Z, LAC-Y, LAC-A. LAC-Z codes for a beta galactosidase, LAC-Y this gene codes for beta galactoside permease, and the LAC-A actually codes for a beta galactoside transacetylase. So these actually the three enzymes which help in transferring the lactose from outside to the inside of the cell, and later metabolize that lactose into glucose for to be used by the bacteria in absence of glucose. So let's move to the how this LAC operon work. I have given you a brief outline what's the structure of this LAC operon. Now moving towards how this LAC operon works. So it's an interesting that there could be four conditions because we already discussed it, that it's due to the presence and absence of both glucose and lactose, which actually on and off this LAC operon. So we have this, it's a simple diagram that this is the LAC-Z gene, structural genes. After Z there would be a LAC-Y and LAC-A. This is the operator sequence, and before that there is a RNA binding site, promoter sequence, and a cap site. So RNA start site for RNA synthesis is from here, and this is one base pair from this. Okay, so there are four conditions that we have both glucose and lactose, ample glucose and lactose available, glucose within the cell and lactose outside the cell available with us to be used by the bacteria. If there is ample glucose available, certainly this operon would be off because there is no need of lactose, and microorganism would conserve the energy. If the other condition is that we have the glucose and there is less lactose outside the body, outside the cell. In both these conditions, when there is ample glucose available, this repressor protein would remain attached to the operator sequence. What repressor protein is, basically this repressor protein actually stops the transcription. So in the initial case, when there is glucose plus lactose are both amply available, the operon would remain off because the cap protein, which is actually attached to the cap site, would not be available. While in the other case, when there is lactose is not available but glucose is available, the repressor protein would remain attached to the operator, and this would actually inhibit the attachment of the RNA polymerase and further transcription. So again, in this case, when there is ample glucose available, the operon would remain off. Keep it in mind, whenever there is a lactose is available in the medium, there is another isomer or its related compound that is called allolactose is also available in the medium. Allolactose has an affinity with this repressor, and whenever lactose is available, allolactose is also available in the medium, and allolactose will attach to this repressor protein and remove this from the operator region. So whenever there is lactose, this repressor would not remain attached to the operator. But for working of this operon, we both need the availability of the cap protein as well as removal of the repressor. So it would turn on only when this repressor protein is removed and the cap protein is attached to the cap site. So the next condition is when there is no glucose and no lactose. When there is no lactose, certainly repressor would remain attached to the operator site, but glucose is absent, so cap protein would be available along with the cofactor CEMP. So once the CEMP and cap is available, it would attach to it, but as lactose is not present, so allolactose is not present and repressor is attached to this point, so operator is blocked and there is no possibility that RNA polymerase could attach and transcribe this operon, and so the operon will remain off. The last condition is when there is no glucose but we have lactose, so CEMP would also be available, and in presence of lactose, allolactose would also be available, so it will remove this repressor from here, the operator site, and RNA polymerase could transcribe and make this operon on an RNA would be transcribed. So this is how the lac operon works on. So this is the representation slide, that how the DNA plasmids replicates within the bacteria. There are two possibilities, that plasmids replicate extracellularly, that it's remain outside the nucleus and replicates and divides into the two cells, or it integrates into the nuclear DNA and replicates with the nuclear DNA. So to end with, in this presentation or lecture, we have understood the basic concepts and terminologies of cloning and recombinant DNA technology. We have realized the importance by discussing the different examples of insulin and growth hormone, how this recombinant DNA technology help us to generate these biopharmaceuticals. And we also recognize the function of expression vectors and the different elements present within the expression vectors, how they help in expressing the transgene. So this is all from my side for today, and we will be having a nice lecture tomorrow.